Hi science friends, welcome back to our final week of cellular respiration, and then we'll jump into photosynthesis after this. So let's go ahead and review a little bit from last class when we talked about the Krebs cycle. So it says walk through the Krebs cycle stating the reactants and products and where they came from or where they go to. So in the Krebs cycle, we have acetyl-CoA that's coming in. Remember, acetyl-CoA came from the pyruvate oxidation step, which also happened in the mitochondria, and we converted pyruvate, which came from glucose. Remember, glucose is a 6-carbon molecule broken down into 2 3-carbon molecules of pyruvate, and then that pyruvate loses carbon dioxide and becomes acetyl-CoA. So acetyl-CoA is what comes into our citric acid cycle. Okay, so let's draw a little cycle here. And so that acetyl-CoA binds with oxaloacetate. I think I missed an O there. Oxaloacetate to form citrate. And then it goes through a series of oxidation and reduction or redox reactions until it gets back to oxaloacetate. So in the process of this, we release NADH, we release CO2, we release FADH2, and we release ATP. Okay? And we talked about that last class. So there's 2 CO2s released, 1 FADH, 3 NADH, and 1 ATP per acetyl-CoA. So that means per glucose, all of those numbers are doubled, okay? And so that's what we've got here. So once we have ended the Krebs cycle, we have only made 2 ATPs from Krebs and 2 ATPs from glycolysis. And so today we are going to get into the final step, the electron transport chain, and talk about where most of the ATP is actually come from. So remember, we only have 1 here. We had 2 in glycolysis. We actually have 2 here because this happens 2 times per glucose. So 4 ATPs thus far before we get started. Just to finish out this question, NADH is going to go to the electron transport chain and drop off those electrons, okay? FADH2 is going to drop off some electrons. CO2 is released as a product of cellular respiration. That's the carbon dioxide that you exhale is coming from CO2 made here and then also made in pyruvate oxidation, and then the ATP as we already mentioned. Okay, so let's get on into today's lesson, which is about the electron transport chain. So the electron transport chain is happening in the membrane of the mitochondria in eukaryotes, and in prokaryotes it will be happening in the cell membrane. So let's just draw a picture of the mitochondria to review. So the mitochondria is a double membrane structure. So this would be considered the inner membrane. This would be considered the outer membrane, and this area in the middle is the matrix. So that's where everything that we have talked about thus far has happened. It's in the matrix, or that middle section of the mitochondria. Now, we've talked about surface area to volume ratio and efficiency, and so this surface area, these folds in the matrix of the mitochondria, are another example of how we're going to add more surface area in organisms and living things to make processes more efficient. So we have extra space for cellular respiration to happen because we have a little extra surface area than we would if that were just a nice little oval or round structure in there. So that's the matrix of the mitochondria. So all of this is happening in this middle space here, okay, between the inner and the outer. Sorry, I kind of drew that backwards. Right here, across this membrane, between the mitochondrial matrix and the intermembrane space. This part here is called the inner membrane space, okay? So everything we're talking about today is happening right here in that barrier. So the electron transport chain is made up of a series of cytochromes, and that's what these little guys are that you see here. Now, some of them have Weird names, okay? But some of them here, these ones that I'm circling now, cytochrome B, cytochrome C, cytochrome A, cytochrome A3, are electron carriers, and they work in the electron transport chain. But what they are is they're proteins with a heme group attached. And so some of these you see have a name of Fe. So what a heme group is, it's a group that contains iron, which is why you see Fe. Iron on the periodic table is abbreviated as Fe. Similar to as in hemoglobin in the blood. Hemoglobin that's in the blood cells is responsible for binding oxygen. It's highly electronegative. And iron, if you remember it on the periodic table, it's a transition metal, so it has a positive charge. So it's going to strongly attract to these negative charged electrons. And so that's what's going to happen here. These cytochromes that are made up of these iron-based heme groups are going to pick up the electrons in the electron transport chain and shuttle them through the electron transport chain. And so NADH is going to drop off these electrons that it picked up. Remember, it picked up the electrons, was NAD, picked up 2 electrons and a hydrogen, and became NADH. Same thing with FADH2. It picked up 2 electrons and became went from FAD to FADH2 and hydrogens. So you may be asking, like, where are these hydrogens coming from? Hydrogen is an ion that's it's present in the cell. So we're going to talk about hydrogens a lot today in our lesson, and so they are present throughout the cell. Hydrogen ions are, and we're going to use them today to do a lot of work. Okay, and so when these electron carriers drop off their electrons to the electron transport chain, they give them to these iron, positively charged iron protein complexes that are going to readily accept those electrons. And ultimately, it's getting passed down here to oxygen. So remember, we talked about before, oxygen was the final electron acceptor, because also it is even more electronegative. And it's going to accept those electrons to be able to form water. What if you remember from your cellular respiration equation, CO2 and H2O. H2O was the other product of cellular respiration. And so that's where H2O is made. CO2 is made in pyruvate oxidation and in the citric acid cycle, or the Krebs cycle. And water is made in the electron transport chain from oxygen, which was a reactant. We had to have that, okay, to do aerobic respiration. But it's being used to make water here, okay? So one thing I want you to notice in this diagram, we really didn't talk about it actually, but there's a difference between NADH and FADH2, and it's where they drop off their electrons. So in this electron transport chain, this represents a series of redox reactions. So again, we've got those oxidation and reduction reactions happening through here. And so NADH drops off higher in the chain. And so what that means is that these electrons are going to fall further, okay? And so they represent more potential energy than FADH2. FADH2 drops off second in the electron transport chain at a lower spot, and so they're not going to fall as far. And so as these electrons are moved through the electron transport chain, they generate energy, okay? But now this energy is going to kind of be used, so it's not actually making ATP yet. It's going to be indirectly used to make ATP, all right? So let's see how that's done. So it's going to be used in this process called chemiosmosis. So that's why we kind of get this name, electron transport chain / chemiosmosis, is really how the book, the Campbell textbook is what, we don't have a set of Campbell textbooks in our classroom, but Campbell textbook is a textbook that a lot of colleges use, but a lot of AP teachers use as well as their basic textbook. And so Campbell refers to this as the electron transport chain / chemiosmosis. And so that's why I kind of call it as such. Most people probably refer to it truly as just the electron transport chain. And then chemiosmosis is kind of a process that happens within the electron transport chain. So what chemiosmosis is an energy coupling mechanism. Now, we talked about energy coupling just a little bit in biochemistry or cells. I don't remember exactly where, but it's basically where you use an endergonic process to drive an exergonic process, or vice versa. Okay, you have one process that's used to drive another one. And that's what's going to be able to use here. That's what's going to happen in chemiosmosis. So remember, osmosis, when we talked about osmosis, it was water moving down a concentration gradient. Okay? Now, I'm going to come back to a chemiosmosis a little bit here in just a minute. So don't get too tore up if you don't fully understand that yet. But we're not going to move water down a concentration gradient. What we are actually focusing on instead of water is these hydrogen ions that I've just been mentioning. So we've got a hydrogen ion gradient, and it's going to drive cellular work, okay? And so when we're here in the intermembrane space, okay, when we move these electrons down the electron transport chain, they are going to drive these hydrogen ions through an enzyme, this enzyme here, called ATP synthase, okay? So we know it's an enzyme because it has that ending ACE, and so it's going to make ATP from ADP, as you well probably figured. But the way ATP synthase works is it's kind of like a turbine. So in a turbine, it uses the wind to make energy, right? The wind spins the turbines, and that spinning of the turbines causes the generation of energy. And so what we're using here instead of wind is we're using these hydrogen ions. And so these hydrogen ions move through this turbine, essentially this ATP synthase, which has a rotor. And as it does, it turns and causes this the conversion of ADP to ATP. Okay? Now, you see right here this PI. That's that inorganic phosphate. So we didn't steal it from a substrate. So this is where we have that oxidative phosphorylation. Okay? Oxidative phosphorylation. So the PI, meaning the inorganic phosphate, is available in the cell, and it's going to be added to the ADP to make ATP. And that's what's going to happen in chemiosmosis. Okay? Now I jumped ahead. I'm going to jump back in just a minute, so hang with me if you're still a little lost. Hang on, we're going to get there. Okay? And so this produces what's called a proton motive force. And so the proton is really referred to as H+. H+ has got one proton because it's hydrogen, and hydrogen has one proton, and so it's referred to as a proton motive force. It also has no electrons because it has the positive charge. Normally, a neutral hydrogen has one electron, but because it has positive charge, it means it's lost an electron, and so truly it's just H+ ion is just one proton. That's really all that's there. And so it's called the proton motive force. So we build this H+ gradient and use it to make ATP, okay? And that's referred to as the proton motive force. So what we've happened here, I want to go ahead and label my diagram. Notice here that the yellow is the matrix and the orange part of the diagram is the intermembrane space. So I'm going to label this the same way, matrix and the inner membrane space. Okay, so here's our electron transport chains. We've got all these proteins, all of our cytochromes. Remember, NADH is getting dropped off first. It's converted to NAD+, and these electrons, the energy that's released there, those electrons represent potential energy, okay? These electrons represent potential energy that was harnessed from the glucose. All this time, we've been breaking down glucose. We break down molecules where we release electrons. We break down a molecule, we release an electron. We break down a molecule, we release an electron. And so these electrons are from our food or from that glucose molecule that we started out with, but they represent potential energy. And so as the NADH is converted to electrons, and these electron electrons move down this electron transport chain. They provide the energy that's needed to move hydrogen ions against the concentration gradient, okay? Down here is low hydrogen ion. Up here is high hydrogen ion, okay? So when we're going from low to high, that's got to be active transport. It's got to have energy, and that energy has to come from somewhere. And that energy is coming from these electrons that are being carried down the electron transport chain. Okay? It's an important concept. And so these hydrogens are moving from low concentration to high concentration, okay? So now we're going to have a buildup. We're going to have all these hydrogen ions up here, okay? Well, naturally, what do these hydrogen ions want to do? Which way do they try to simple diffuse? Use simple diffusion. Which way do they go? Well, they like to go this way, okay? Now, they can't go this way through these proteins. These protein channels are one-way paths, okay? They are usually not two-way streets, okay? But what does allow a channel back down this way is ATP synthase, okay? And so when these hydrogen ions move through ATP synthase, they cause that turbine to spin, which results in the conversion from ADP to ATP, okay? And that's what chemiosmosis is, all right? We've got energy coupling here. We use this process, which absorbed energy. This was an endergonic process. It absorbed the energy. Sorry, I'm a little sloppy here. It absorbed the energy from those electrons as they moved down the electron transport chain, okay, to drive an exergonic process. And this is exergonic because it's making ATP, okay? And that's chemiosmosis. So this first part is the electron transport chain. These electrons moving down this chain, which produces the energy that causes this second step, which is chemiosmosis. And so the book describes them as electron transport chain and chemiosmosis together make up oxidative phosphorylation, okay? And so that's how that works, all right? We've got our oxygen here that pick up these electrons at the end of the cytochrome chain and convert it to water, okay? And like we said, that was one of our products. But that is the electron transport chain, chemiosmosis, and it's oxidative phosphorylation because of the inorganic phosphate where it came from, where the ATP, the P for the ATP came from, okay? That is that. I think this is lovely. Y'all probably think I've lost it. All right, so let's look at this big picture and then we'll go back and draw some stuff, all right? So remember, in our first step, glycolysis was happening in the cytoplasm, okay? We took our glucose molecule, our 3-carbon glucose, C6H12O6, we broke it down into pyruvate, okay? In the process, we released some NADH, which is ultimately going to the electron transport chain, okay? And we released 2 ATPs, all right? Those pyruvate were shuttled into the mitochondria for pyruvate oxidation. Honestly, that energy there also is coming from the electron transport chain. That's kind of where that's coming from. And so pyruvate is oxidized, means it lost electrons and became acetyl-CoA. In this process, we also gave off CO2. All right. And some NADH was released. I think before I said maybe there wasn't NADH released, but I guess there is. I don't think that's critical whether you know that there was some release there or not. It wasn't on any of the other diagrams, but it is on here. So I would have thought there was because the broke bond down and you've got electrons, so they had to be picked up. But it wasn't on any of the other diagrams. But I'm going to go with it's made there, okay? So you won't be tested on that. Obviously, AP is not going to be that picky about is NADH made in pyruvate oxidation, so don't worry about that. And so We've got these NADHs, these FADH2s that are going to be made. Remember, they are all going down here to the electron transport chain. You've got some more that are going to be made in the citric acid cycle. Citric acid cycle is also going to release 2 ATPs. It's also going to release CO2 again as well. Okay? And then finally, though, this last step, this oxidative phosphorylation and electron transport chain, chemiosmosis, whatever you want to call it, it releases about 26 or 28 ATPs. We don't really have an exact number. If you read different textbooks, you would get different answers as to how many ATPs are made. And it's going to be a little bit different from like cell types to cell type too. So in some cells, you have more NADH. In some cells, you have more FADH2. And so remember, because NADH releases it a little bit higher up, it's going to make more energy than FADH2 is going to make a little bit less energy. So these numbers are fixed. 2 and 2 here, we know for sure. This number is a little bit questionable. Okay, but ultimately the range you usually hear about is between like 28 and 36. I've never honestly heard anything less than like 28 to 36 ATPs. So just know that most of it is truly coming from this oxidative phosphorylation. Don't worry about the total number. Just realize it's a lot more. You get 2 and 2 and like 20 something, okay, or 30 maybe even. But about 28 to 36, 30, 32, whatever number ATPs are going to be produced total in cellular respiration per molecule of glucose. Okay, so cellular respiration is about 35 to 40% efficient as far as how much energy is actually available in glucose and how much energy is lost to heat. So even the most efficient cars are about 25% efficient. And so it is considered to be a metabolically an efficient process. Although that number sounds low, 36% efficient, you're like, crap, that's not very good. But metabolically, it is considered to be a pretty efficient process. Okay? So let's draw just a little bit to kind of make this our own. And we're going to start from the top and draw. So step 1, we've got glycolysis. Okay, remember it's happening in the cytoplasm. And so we have our glucose, our C6H12O6, and we're going to break it down into 2 pyruvate molecules. Okay, in step 2 is pyruvate oxidation. It's going to happen in the mitochondria. So some of that energy from the electron transport chain is actually driving that movement to the mitochondria. And these pyruvate molecules are going to be converted to acetyl-CoA. Okay? You're going to lose, in this process, you're going to lose 2 electrons that are going to be picked up by NAD+ to form NADH. NADH is going to the electron transport chain. Okay. Here, same thing as well. You're going to lose 2 electrons that are going to be picked up by NAD+ to form NADH. That's also going to the electron transport chain. You're also going to release in this one 2 ATPs. Now this step is not going to release any ATPs, okay, in the pyruvate oxidation step. All right, so now this acetyl-CoA is going to enter the Krebs or citric acid cycle. And it's going to join with oxaloacetate. Okay, and this is step 3. This is also in the mitochondria. The matrix. This is the matrix too. This step is also going to release CO2 also, sorry. This step is going to release CO2. This one's going to release 1. This one's going to release 2 CO2s. It's going to release 1 NADH, 3 NADH, sorry, 1 FADH2, and 1 ATP, okay, per oxaloacetate. And remember, you're going to make 2 of them, 1 from each of the pyruvates. Okay. And then, of course, these are going to go to the electron transport chain. This is going to be a product of your respiration, as is the CO2 here. Okay. And in this 4th step, I want to leave plenty of room for this 4th step. We have the electron transport chain. So again, this is happening in the mitochondrial membrane. Okay. So in our membrane, I'll draw our membrane here, we have these hydrogen ions in the cell. Okay? And they, as these electrons are being dropped off, we've got NADH that's dropping off electrons. We've got FADH2 that's dropping off the electrons. Okay? These electrons are producing energy. Whoops, I draw my arrow the wrong direction. These electrons are producing energy that's going to cause these hydrogens to be moved out of the cell. Okay? And so this represents active transport, because we have low hydrogens here and high hydrogens here. And so they're moving against the concentration gradient. So we see all these hydrogens that are building up here, more hydrogens on the outside than there are on the inside. And they want to get back through. And so their way back through is through ATP synthase. And as they go back through, they will cause ATP to be converted to ATP. And that represents our oxidative phosphorylation. Okay? This part is the chemiosmosis. This part is the oxidative phosphorylation. All right. The chemiosmosis means the energy gradient. We're using this hydrogen gradient. We're moving hydrogens to be able to use energy coupling to allow for the production of ATP. All right. So hopefully that makes sense. If y'all have any questions about it, please let me know. Remember, this step has to have oxygen. Oxygen's really important because it gets these electrons at the end and is used to form water, which is another product of cellular respiration. And so oxygen is the final electron acceptor. Tomorrow, we're going to talk about anaerobic cellular respiration. So anaerobic respiration is what happens when there's no oxygen, okay? So just to kind of, since we're on the electron transport chain right now, I'll just go ahead and give you a little preview. If this oxygen is not present, then all of these electron carriers get backed up with electrons. They can't hold any more electrons. So guess what's stuck? NADH and FADH2 get stuck. They can't drop off electrons because there's nobody they can give them to. Nobody's there at the end of the assembly line to grab those electrons because there's no oxygen here, okay? And so as a result, NADH and FADH2 get backed up. And so the other stages of cellular respiration, here that relied on NADH, and here that relied on FADH, and here that relied on NADH, and here that relied on NADH, those can't all happen, okay? If there's no oxygen, then the whole process gets backed up. And so the previous steps really can't happen because NADH and FADH2, remember, they're recycled. After NADH and FADH2 drop off their electrons here, they go back to being FAD plus and NAD plus, and they go back and start the cycle over and pick up more electrons. But if they can't drop them off, they can't be recycled, okay? And so that's going to cause some problems. And tomorrow we'll talk about anaerobic respiration and what happens instead, okay? So this wraps up aerobic cellular respiration in a nutshell. If you have any questions, please reach out and let me know. You have your respiration worksheet to finish today. I think it's Model 3. And then you can go ahead and submit that. And then tomorrow we'll talk about anaerobic respiration. And then the next day we will jump into photosynthesis. We're going to wrap this up, take a test next Thursday before we leave for Christmas break. And then next Friday, actually, there will be another review quiz for the whole semester. So we're kind of reviewing some of this stuff as we go along. We've hit a lot of biochemistry in this unit. We've hit a lot of the cell stuff in the transport. We've hit some of the organelles with the mitochondria and different things. And so we're going to go back and review some of those at the end of the semester as well, some of the major big points. So if you have any questions, let me know. And if not, then have a great day.